Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A Level Biology Unit 5 for January 2021. I will do this paper in two parts, so I'll make two videos. The part 1 video will contain question 1 to question 4, and the part 2 will contain question 5 to question 8. I will put the link to the part 2 video below the description box of this video. And please do not forget to subscribe to our channel. So let's begin. Question 1 says, nerve impulses travel along axons as action potentials. The graph shows an oscilloscope trace for an action potential in an axon. So here on the vertical axis, we have potential difference in millivolts, and then on the horizontal axis, we have time in milliseconds. So the first question says, which is the resting potential for this axon? The resting potential, of course, is about that, which is negative 70 millivolts. The answer should be A. Let's go to the next part. Here they say, both voltage-gated sodium and voltage-gated potassium ion channels are involved in generating an action potential. Which of these voltage-gated channels are open at Y? I want to take you back to point Y, just a minute. Y is this point here. So at that point, let's continue down. Y is, let's see, they said no voltage-gated ion channels are open. That is wrong. And here voltage-gated sodium ion channels are only open now. It should be the voltage-gated potassium ion channels only. So in this case, the answer should be a C. So the next question says, which is the state of polarization of the membrane at Z? Let's go back at Z and see. Z at this point, this is, of course, as you can see, the potential or potential difference is increasing. So that part is hyperpolarization at B, at that point Z. So the answer here should be a B. Moving on, they say, how many of the following statements are correct? Here we have, the magnitude of the action potential is proportional to the strength of the stimulus, that generates the action potential. Not necessarily. Action potentials spread out in both directions along the axon. No, they do not do that. Following an action potential, there is a refractory period during which it is not possible to generate a new potential. This is correct. So the answer should be only one is correct. There is a refractory period before the next action potential is generated. So the answer is B. Explain why maintaining a resting potential requires ATP. So remember, to maintain a resting potential, the sodium-potassium ion pump has to pump sodium ions outside and potassium ions inside. To do that, energy is required. So I say the sodium-potassium ion pump uses ATP to pump sodium ions out of the cell or out of the axon or through the membrane, and the potassium ions into the cell across the membrane. This maintains a concentration gradient for sodium and potassium ions, which gives the membrane its resting potential. So we are looking at a way of maintaining the resting potential, and that is how it's done. This brings us to the end of question 1. Let's move on to question 2. Question 2, many athletes train to improve muscle strength. In an experiment, the effect on a muscle of 12 weeks of low or moderate training was investigated. The graph shows the effect of 12 weeks of training on muscle strength and muscle cross-section area. So we have two graphs. The first one has the percentage increase in strength against the low level of training. So we have low and moderate. And here we have increase in cross-section area, and then against level of training, which is also lower to moderate. So continuing on, they say, the muscle strength of the athlete before moderate training was 12.25 AU. Calculate the muscle strength of the athlete after 12 weeks of moderate training and give your answer in 3 significant figures. So here we can see, this is, since they are looking for muscle strength, we're going to use this graph here, and we can see after 12 weeks of moderate training, the percentage increase is about 27.5, looking at that part here. So I call this 27.5, and since this is the percentage increase that has occurred, it means we're going to use the original muscle strength that was there before the moderate training and use it to calculate the muscle strength after the 12 weeks. So here I say, if we have, this is the original, to find the increase in strength, we will say percentage increase times the original strength, and it gives us this. So this is the increase 3.4375 AU that has occurred. And to find the muscle strength after, it should be the initial muscle strength before the training plus the change that has occurred. So you can see the strength before the training, which is 12.5, plus the increase in strength, which is 3.4375. If we combine them together, we get 15.9 a minute. Let me try to change this. Yeah, it will be 15.9375, which is 15.9 AU. So adding up, and this is going to be the answer. The key thing is you knowing that the percentage increase was this. If this is the percentage increase, which is 27.5, divide by 100 and multiply it by the original before the training, which gives us the change. And then the change that has occurred plus what was there originally gives us the total that is present right now. So the answer is 15.9. Let's move on. B, the type of muscle fibers present in samples taken before the training and after 12 weeks of low and moderate training were determined. So this table here shows us the slow twitch before training as well as after 12 weeks of training, the percentage of the fibers that are present. So this is percentage of slow twitch, the percentage of fast twitch, and the percentage of intermediate twitch. We can see before training and after training whether low or moderate, the slow twitch are exactly the same percentage. However, when we come to the fast twitch, we can see here there is a decrease, minus 4%, and here we can see minus 1%, while here we see there is an increase of plus 4%, and here we see an increase of plus 1%. So we can see that the magnitude of change occurring here is the same as that occurring here, but this is decreasing as this is increasing. This is evident that 4% of the fast twitch are going to be converted into intermediate twitch, and here we can see 1% of the fast twitch will be converted into intermediate twitch as well. So we can see the key information from here, or what we can observe, is that as intensity of exercise increases, fast twitch muscle fibers, some of the fast twitch muscle fibers are going to be converted into intermediate muscle fibers. So let's continue. So they say intermediate fibers have structural properties between those of slow and fast twitch fibers. Describe how the structure of an intermediate fiber will differ from the structure of a slow twitch fiber. Remember, slow twitch fibers are for endurance, so they will have more mitochondria, a lot of myoglobin, and so on. So it means intermediate fibers will have fewer mitochondria in comparison to slow twitch, and then they will have a higher glycogen content store, and then they will have less myoglobin as well as more sarcoplasmic reticulum. So if you had written 3 of these, you get your 3 marks. I think that's it. Let's continue to the next part. This part says, comment on the effect of exercise on this muscle and use the information in the graph and the table to support your answer. I want us to go back slowly to the table, and then I'll take you back to the graph. We can see the table. The key information we can see here is the slow twitch did not change. So it means slow twitch muscle fibers are not affected by exercise. Whether you use low training or moderate training, they're almost exactly the same. The percentage does not change, yet fast twitch fibers are slowly converted into intermediate fibers because there is a decrease here and an increase of the same intensity. Also here we see 2 to 1, 54 to 55, we see there is the same thing. So with intensity or with increase in intensity of exercise, fast twitch are converted into intermediate. Let's go to the graph. The graphs we can see as well, there is a percentage increase in strength. We can see a moderate training, there is a higher percentage in strength than low training. For both graphs here, we can see there is increase in cross-section area with moderate training in comparison to low training. So we're going to use these and be able to answer the question on the other page. So here I say, exercise has no effect on slow twitch fibers, and also training leads to greater muscle strength and cross-section area. We can see this part from the graphs and this part here from the table. Training causes fast twitch fibers to become intermediate fibers. We can see that change from the stuff I explained from the table. This is what I explained there. And then the greater the level of exercise, the greater the switch from fast to intermediate, basically. So this brings us to the end of question 2. Let's move on to question 3. Question 3, the brain is responsible for processing information in coordinating responses. The diagram shows a human brain. 3 regions of the brain have been labeled. So here we have region Q, region P, and region R. The first question here says, complete the table with the name and function of each region. So region P here, of course, this is the medulla oblongata, and it's responsible for non-conscious regulation of body processes like the heart rate, as well as the breathing rate. Region Q, which is that one here, is the hypothalamus and is responsible for homeostatic control like thermoregulation as well as osmoregulation. And then R here we have the cerebellum, which is responsible for balance and coordination of movement. Let's move on. B says, seizures in the brain can be investigated using different imaging techniques. Which one of the following statements is correct? CT scans are produced by the absorbance of magnetic fields. That is wrong. CT scans do not use magnetic fields, they use X-rays. Part B says CT scans are produced by the absorbance of radio waves by deoxyhemoglobin. That is wrong. C says CT scans produce a cross-section image of a thin slice through the body. That is okay. And D says CT scans can be used to study brain activity while a person is undertaking tasks. That is wrong. So the correct answer here is a C. Roman 2 says, during a seizure, part of the brain will be overactive. Describe how positron emission tomography could be used to identify the part of the brain involved. Again, remember, with positron emission tomography, the region of the brain that is hyperactive is going to take up glucose and carry out respiration, and therefore they can be able to trace which part of the area is actually overactive. However, to do this, they have to use an isotope that can emit radiation in order for that radiation to be tracked. So here I say, part of the brain that is overactive will have overactive neurons, or it will be basically the neurons or the nerves in that area are going to be taking up more nutrients in order to provide ATP through respiration. So these neurons will require an increased supply of glucose for ATP supply. So incorporation of positron-emitting isotopes, example carbon-11 or carbon-15 into glucose, makes this glucose to emit gamma radiation or gamma rays that are detected and converted into an image. So these can be observed during activity. So they can know which regions of the brain are being overactive and therefore taking up the isotope which is contained within the glucose. So that will be okay. Remember, glucose contains carbon 6, hydrogen 12, or 6. So some of these atoms, like carbon atom, you can use carbon isotope 11, or you could decide to use oxygen isotope 15. These are going to be emitting radiation, so you can be able to observe. It can be, maybe you can use only carbon, which with isotope 11, or you could just use oxygen 15, and that's it. So these can emit radiation, and therefore you can be able to trace which regions are being overactive, and therefore create an image that is required. Moving on. Here they say, some seizures can result in an increase in body temperature. Again, remember, seizures are these occur in regions that are very active, and body temperature. So very active regions are going to require a lot of glucose, and therefore they'll respire a lot, produce a lot of ATP. So this ATP produced, some of it is going to be lost as heat, and when it's lost as heat, the body temperature is going to increase in that region, leading maybe to an overall increase in the whole body temperature. So they say, suggest how a seizure in part of the brain could lead to an increase in body temperature. Here I say, increase in the impulse that arrive from the neurons at the seizure site causes more electrical activity or impulses to arrive at the thermoregulatory center in the hypothalamus. This causes an increase in the metabolic rate. So here we can see more respiration of glucose, higher production of ATP, and some of the ATP is lost as heat, hence the increase in body temperature. This is a 3-mark question, and writing all this would be able to get you the 3 marks you're looking for. So this brings us to the end of question 3. Let's move on to question 4. Question 4. Skeletal muscles function to move the limbs of an organism. The diagram represents a sarcomere from skeletal muscle. So we can see here, this region here is a sarcomere. So they say which protein forms the structure labeled X. Structure labeled X is the myosin protein. Of course, we know structure Y is the actin, so this is the myosin in that region. The answer here should be B. The next part says, which part becomes shorter when the muscle contracts? Now let's see. When the muscle contracts, you remember these actin filaments are going to pull onto this side like this, as the actins are pulling onto this side. So this region here is the one that is going to decrease. So here we can see the answer should be a D, which is region Z. Moving on. How many of the following statements are correct when a sarcomere contracts? During the contraction, calcium ions bind to troponin. That is correct. Calcium ions are released from the sarcoplasmic reticulum. Correct. And then myosin changes shape. That is also correct. So here we can see the answer is D because all three statements are correct. The next part says, which molecule contains an enzyme that hydrolyzes ATP? You guys know ATP is found on the myosin head, so it should be on the myosin. The answer here should be a B. Let's move on to the next part. Part B says, inherited mitochondrial disease affects muscle function. The diagram shows the electron transport chain in a mitochondrion. So here we can see there is complex 1, whereby reduced NAD comes and leaves oxidized. And then here we have complex 2. We have reduced FAD, leaving as oxidized FAD. And then we have COQ, and then complex 3, we have cytochrome C, and complex 4. In cytochrome C, we can see there is reduced TMPD, which leaves as oxidized TMPD, because the reason as to why this leaves as oxidized because it has reduced this. So basically when this comes in, it reduces that, and then it leaves as oxidized. So in this case, we can see that these carrier molecules that reduce NAD, reduced FAD, and reduced TMPD, they are going to reduce the complexes within the electron transport chain. Another thing we have to observe is we can see this pathway here, can you see, to complex 1 directly links to COQ, not to complex 2. So meaning if there is a blockage in complex 2, the electron transport chain can still function. If there is also a blockage in complex 1, reduced FAD can still come and reduce complex 2, and the electron transport chain can still function. However, if there is a blockage in complex 3, everything is going to be stopped. If there is a blockage in cytochrome C, everything is going to be stopped. And if there is a blockage in complex 4, everything is going to be stopped. So this, that, and that, mainly this one and that one, they're very important. So if there is a blockage here, provided this is functioning, it's okay. If there is a blockage here, provided this is functioning, it can still be okay. So let me move on. They say, in an investigation, samples of muscle tissue from healthy individuals and from 4 individuals with mitochondrial disease were collected. The function of mitochondria when these samples were treated with different substrates was investigated. Treating muscle tissue from healthy individuals with pyruvate and malate increases the production of reduced NAD. Succinate increases the production of reduced FAD, and reduced TMPD increases the reduction of cytochrome C. So you can see that. So they say the oxygen consumption of the muscle tissue samples from healthy individuals and from 4 individuals with mitochondrial disease were recorded. The results are shown in the table. Please ignore the marks I put here. I did that for the explanation on the next page. However, I'm going to try to talk about the observance here. Keep in mind that we are going to use the information from the healthy individuals as being normal. So the normal oxygen consumption when treated with pyruvate and malate should be between 16 to 26. I'm taking this plus or minus 5, so it gives me this range. And for this treating with succinate produces, it should be it should be between a 9 to 18 plus or minus 4.5, and this one should be between 43 to 83 plus or minus 20. So when we look at this table, we can see individual 1, their oxygen consumption is not within this range, so they're out. Individual 2, we can see it's within the range, so that is okay. Individual 3, not, and individual 4 is not because they are far away from this range. Trading with this, we can see individual 1 is out because they are not in this range. Individual 2, 10 is within the range, and individual 3, 14 is within the range. And individual 4, 6 is not in the range. However, remember 6 is kind of closer to 9, so they are somehow, but not within the range of being exactly normal. Now, with reduced TMPD, individual 1, the range should be between 43 to 83. So that is out. That is within the range, closer to the range. So I take that and take out that. So knowing all this information is going to help us answer the next question. So let me move on. So here they say the results of this investigation can be used to identify the defects in the electron transport chain of individuals with mitochondrial disease, comment on the defects in the electron transport chain in the individuals with mitochondrial disease. So the first part I wrote here was to explain what you remember they are treating with pyruvate malate, succinate, as well as reduced TMPD. I began off by explaining what these molecules or substances they are treating the individuals with, how do they affect the electron transport chain. So I say pyruvate and malate lead to production of reduced NAD. And you guys saw from the ETC, reduced NAD is going to have its effect on complex 1. So it means there is going to be more oxygen consumption by complex 1 if that and that are released. So also treating with succinate increases production of reduced FAD. And if we look back here, we can see reduced FAD has its effects on complex 2, meaning it's going to make complex 2 take up more oxygen or consume more oxygen during this process. And then reduced TMPD causes a reduction or more reduction of cytochrome C. So if no more oxygen is consumed, there is no defect in that cytochrome C. Then I went on to talk about the different individuals. So I said for individual 1, all treatments did not stimulate normal oxygen consumption. Again, I want to take you back slowly here. You can see from the table, individual 1, everything, I crossed everything out because for all treatments, the oxygen consumption was not within the required range. And you can see for individual 2, they're all within the required range. Individual 3, we can see only this and that are within the required range. And for individual 4, this is not, that is not, but this is close to the required range. So we can call it kind of normal because 40 and 43 are closer to each other. So here I went on to say individual 1, all treatments did not stimulate normal oxygen consumption. Then the defect in this individual, that is individual 1, is in or after cytochrome C. Because again, to take you back, you can see if this is not functioning well, that is not well, and that is not well, you can see it should be within cytochrome C. Maybe there is a blockage here. Remember what I told you, that if there is a blockage here, it's going to stop everything else. If this is not this is not consuming oxygen in the required amount, and this is not doing the same, and that is this is not doing the same, this one here, it means the blockage is either in here or after. Because if this wasn't functioning, this would function and the ETC will not be blocked. So any blockage is because the problem is here or after this part. So let's move on back to where we were. Let's get to individual 2. So for individual 2, all treatments lead to normal oxygen consumption, so that effect is before complex 1, since there is no fault in the ETC. So this person has mitochondrial disease, yet the ETC is okay. It means the problem is before the electron transport chain, or before complex 1 of the ETC. So for individual 3, only succinate and reduced TMPD stimulated normal oxygen consumption, so that effect is in complex 1. Again, to take you back, if this is okay, if this one here is okay, and this one here is okay, then it means the problem is here. So it's complex 1. They have the mitochondrial disease because complex 1 is not functioning properly. And lastly, we go to individual 4. Only reduced TMPD stimulated high enough oxygen. Remember, this was 40. It was not within the range, but it was closer. So they have almost no more oxygen consumption. So this means that effect is either in complex 1, or it can be in COQ or in complex 3. If this is not working well, the problem could be here, it could be here, or it could be here. So that is why I said for individual 4, they will have the problem in that, or that. So this brings us to the end of question 4, as well as the end of the first video. So please do not forget to subscribe, and I'll see you in the next video. Bye-bye.